Thank you everybody for sticking around to the end to hear my talk, and a big thank you to the ANZ S&M conference organisers for the opportunity to speak here today. I'm Simon Puddick, I'm Chief Technology Officer for AdvanCell Isotopes, and it's my pleasure to deliver this talk today on solving the demand for a scalable supply of lead-212 for targeted alpha therapy. A few disclosures up here on the screen, and to say that some of the statements made in this presentation are forward-looking statements. AdvanCell is an Australian radiopharmaceutical company with a platform technology for targeted alpha therapy. We use our technology to make a portfolio of products, the first being alpha-212, which is lead-212 as an active pharmaceutical ingredient. We use this lead-212 to progress 3 drug candidates, ADVC-001, 2, and 3, in metastatic prostate cancer, metastatic melanoma, and pancreatic cancer and breast cancer, respectively. I don't think I'll be the first or last person today to show these images, and to say that it is a very exciting time to be in the field of nuclear medicine. Radioligand therapy is set to be the 4th pillar of oncology. We now have demonstrated outstanding clinical efficacy for 2 indications, and I expect that there will be many, many more. With very heavy buy-in by the major pharmaceutical companies, we do expect this field to grow significantly and very fast, and there are a lot of new targeting ligands that are rapidly being developed, indicating that this method of treating cancer will grow into other areas of oncology. There is a very rapid clinical development pathway for new radioligand therapies because of the ability to image patients before treatment using companion diagnostics. Through this mechanism, the therapeutic path can be visualized in a patient before the dose is administered, and that significantly compresses the clinical development timelines, and again, makes it a very exciting time to be in nuclear medicine. Lutetium-177 really has been the mainstay therapeutic isotope for radioligand therapy to date, and has delivered, as I said, outstanding clinical efficacy. However, there are still significant challenges associated with lutetium-177. There are a lot of patients who either don't respond to lutetium therapy or rapidly progress following lutetium therapy, and there is a need for additional innovation in radioligand therapy to improve long-term survival, but also to provide therapeutic options for patients who do not respond to lutetium therapy. And we believe that alpha therapy is one of those innovations that is incredibly exciting. Another aspect of the challenges ahead for lutetium really is manufacture at scale, and I don't believe that this is currently solved. Both carrier-added and non-carrier-added lutetium-177 require high neutron flux reactors to achieve suitable yields, and those reactors are old. I've just listed a few of the reactors around the world. Actually, this is most of the reactors around the world that produce lutetium-177, and as you can see from the year that they went critical, this is a very aging fleet of reactors globally that produce lutetium-177. The 2 outliers really are Opal in Australia. We're in a very fortunate position, and FRM2 in Germany. But there is going to be enormous demand on this very aging infrastructure to produce lutetium-177 at scale, and I don't believe it's up to the task. In addition to that, non-carrier-added lutetium-177 requires about 1 gram of ytterbium-176 as a target to produce roughly 50 patient doses of lutetium. And there is one supplier of ytterbium-176 in the world, which is Rosatom in Russia, and that supply chain is currently under significant threat. As an alternative to both the clinical efficacy and the manufacturing challenges, alpha isotopes are very exciting. I've listed 3 here, astatine-211, actinium-225, and lead-212, which is where most of the activity is focused at the moment. I've just tried to outline the production methodologies, so the green box going to the purple box, and then the decay chains for each of these isotopes, as alpha decay is a little bit more complex than beta decay. And just highlighting a couple of the properties of these isotopes and how that matches on to clinical potential. Astatine-211 has a very nice half-life for that's well matched to small molecules or peptides, the biological half-life of small molecules or peptides. But the manufacture of astatine is incredibly complex. 28 MeV alpha beams on a natural bismuth target, followed by scraping of that target and sublimation of the astatine, followed by some very complex chemistry to then coordinate the astatine to a targeting molecule. And that in itself, I think, will restrict astatine to centers of excellence who are currently able to produce astatine 211. There is a significant impediment to making astatine at scale. Actinium-225, very clinically exciting, although the decay chain is slightly scary. A 10-day half-life for actinium-225 with 4 alpha emissions and quite a few reasonably long-lived daughter isotopes, which will all undoubtedly be unbound from the targeting ligand following the first alpha decay due to the recoil effect. In addition to that, the manufacture of actinium-225 is significantly constrained. Global manufacturing capacity is currently about 63 GBq per year, which is enough to treat maybe 2,000 patients. And that's via this thorium-229 route. So stockpiles of thorium-229 in the US, Germany, and Russia have the capacity to make about 63 GBq per year of clean actinium-225. Thorium spallation methods, as most people know, are well in development and have the potential for very high yields, but with the associated actinium-227 contamination, which is very, very challenging. In addition, there aren't that many 70 MeV proton beams around. Proton irradiation of radium-226 targets in conventional cyclotrons is definitely being explored and is an exciting option, but manufacturing the radium-226 targets is still, I believe, an unsolved challenge. In complementing all of those things, lead-212, we believe, is the ideal therapeutic isotope for alpha therapy. It has a very nice half-life, 10.6 hours, very well matched to small molecules or peptide targeting ligands. The chemistry of lead is very well known, and there are very good chelators for lead. DOTA itself is a very adequate chelator for lead. There is a single alpha emission via one of the branches from bismuth 212, and so the recoil effect is not really an issue with lead-212. And using generators, there is the possibility for daily manufacture at scale of lead-212. So AdvanCell have developed and built a generator making lead-212 from a proprietary thorium-228 source. The generator operates in a fully automated fashion, can be installed in a hot cell, and flexibly deliver 100 MBq to over GBq amounts in future generations of lead-212 per day on an ongoing basis. Because the half-life of the thorium-228 source is 1.9 years, the lifetime of the generator is very long. It will operate for 12 to 18 months. The generator is assembled, transported, and installed cold. So the generator itself is separate from the source, and the source is manufactured separately, transported separately. A, for ease of logistics, and B, so that replacement of the source is simple. The generator stays installed in a hot cell, and a new source can be loaded in after 12 to 18 months to continue manufacture of lead-212. And we believe that this provides a reliable, scalable, and GMP-compatible solution for targeted alpha therapy. What comes out of our generator is alpha-212. So this is a dose of lead-212 as an active pharmaceutical ingredient, and we are currently manufacturing this in Brisbane. We make alpha-212 on a daily basis, and we have the capacity to dispense over 100 megabecs from our facilities here in Brisbane. We will shortly be producing this for use in clinical trials and other clinical research with the intention of CGMP in the near future. And we're very excited to make this isotope available for research use around Australia. So a few of the details on alpha-212. The dose comes out of our generator in 0.4 ml of 0.05 molar HCl with a pH of between 1 and 2. We can achieve a specific activity of over 5 GBq per micromole. So for a patient dose of around 100 megabecs, this is a good specific activity. We obviously confirm the identity of the isotope by gamma spectroscopy before release using the 238 keV line from lead-212, and we have less than 0.002% radionuclidic impurities in the dose. In terms of metal impurities, these are low, and helps to achieve our high specific activity. A little bit on some radiolabeling experience. So we've now used our lead-212, alpha-212 to label a number of compounds, and what I'm showing here is just some radiolabeling data using dotatate. So labeling with lead-212, this is typically what we see in the HPLC trace and in the TLC for a labeled compound. You get the lead-212 peak just right here, but also you also get the bismuth-212 peak as the active daughter of lead-212. The thallium-208, which is after the alpha decay of bismuth-212, so the recoil effect of that alpha decay, is the recoil energy is easily enough to break the chemical bonds of the chelator, and the thallium then comes free. So we usually see this thallium-208 peak with a little bit of unbound bismuth-212 right at the start of the HPLC trace. We also see this in the TLC, and so this is a way that we can monitor stability by TLC, because any unbound lead-212 comes out at the front, comes out at the top of the TLC plate. So we typically run the TLC, cut it in half, and then have a look at the bottom and the top by gamma spectroscopy, and you can quite clearly see the very large 238 keV peak for lead-212 down the bottom, and an absence of that 238 keV peak. It's this tiny little bump here in the top half, which consists mostly of thallium-208 and bismuth-212. So just to summarize, AdvanCell is manufacturing lead-212 on a daily basis in Australia using our proprietary generator. We're manufacturing over 100 megabecs per day. We are very excited to be able to offer that isotope to the research community in Australia. We will shortly be supplying lead-212 into clinical trials. Our first clinical trial of ADVC-001 we expect to start in 2022, this year, with GMP supply in the near future. Radiolabeling is facile. We get over 99% radiochemical purity. It's efficient, and the stability data is very promising. Please come and talk to us if you're interested in lead-212 or in collaborating, or visit our website, and please do follow our LinkedIn page. We occasionally post company updates there. Thank you very much for listening, and again, thank you to the conference organisers for the opportunity to speak today. Thank you.